If you're that person who just keeps on getting confused between transcription and translation and can't tell the difference, this video is for you, my man. In this video, we're going to talk about what transcription is. We're going to understand why transcription is different from translation, and you will be able to distinguish the two by the end of this video. So we talked about coding RNA in the previous video, and coding RNA is when we have DNA going to mRNA, which is the only kind of messenger RNA and the only kind of coding RNA. And we also talked about non-coding RNA, which really refers to DNA going to other kinds of RNA, which are not directly responsible for the process of translation, but they have an important role inside it. So we are going to talk about what transcription is, and really, if you focus on the word transcription, the word script is inside it. The script really just refers to the writing and copying of what we call the master book, which is the DNA. The DNA is like the master book, and everything has to be copied from it and then sent off. Because we have the DNA sitting inside the nucleus. One, we can't have DNA doing directly going to directly participating in translation, because they're tightly packed up in the nucleus with something called nucleosomes, and we talked about it before. Nucleosomes are 8 histines that are wrap on which these linear DNAs can wrap around, and because DNA is tightly coiled up and dense, and because we have to store it for future use, we can't just have DNA directly doing translation. And because of this, there needs to be a script that is sent out in terms of eukaryotes to the cytoplasm where translation can happen. So transcription is when we have the script being made, which is called the mRNA, the messenger RNA. So before we discuss anything else, I want to ask a interesting question. The question is, let's say that the ambition of a gene is to, or let's say there is a specific gene on this strand over here, the B strand, and let's say that the B strand is we need the B strand to to be copied. So how is this going to happen? So you might be thinking that a an RNA polymerase will come. That's important. That's an important difference right out the bat. You can't say DNA polymerase works as over here. It's RNA polymerase. So the RNA polymerase will come and it will, if you think it will copy this strand over here, that will produce. Now let's think about it logically, right? Let's look at this strand over here, right? If this strand over here is being copied, it's not going to produce this strand. It's gonna produce its complementary strand, and because of this, because of complementary base pairs, if the ambition is to make the B strand, the A strand is going to be synthesized, because think about it, this is the complementary of B, and when this is synthesized, or RNA polymers will be copying this off, it's going to make a complementary strand, which is going to be the B strand. That's a very important difference right off the bat, and I want to highlight this even further on. So let's say that. So I'm going to ask you this question again. Do both templates of, this is another important, so we're going to build onto that thought we just had a second ago. Do both templates of a double-strand DNA code for the same mRNA? And now let's think about this logically. We have a double-strand DNA, and one of them is red and blue to distinguish the two different strands. And let's say that the blue strand has the codons for GGG, 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 and then the red codon is the red strands have a codon for CC, CCC, CCC. So let's say that we have transcription for the blue strand. We can have a cytosine. So this guanine is transcribed into an mRNA. It's going to have a cytosine. And if this cytosine were translated later on, and translation is a process when the mRNA is able to, is able to make polymers, or make protein through translation. And don't worry about translation yet, because we haven't talked about it, but translation really quickly is just the mechanism where the message is translated Exactly, it's translated to the specific amino acid code. Uh, so so when the mRNA is made and is transported to the cytoplasm for the eukaryote especially, it's gonna code for proline, proline, proline. Now, if the opposite strand, which was the red strand, when it had a sequence of cytosine, cytosine, cytosine being repeated, if this same sequence was translated, we would get the opposite guanine, 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 and if this were translated, we would get glycine, 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 and very obviously these two are not the same amino acids produced. So it's important to remember or understand that these two strands have different genetic material on them, and therefore they have different amino acids that will be synthesized. And another thing that I want to highlight once again is if this blue strand over here has a gene that is required, the other strand will be replicated for the very reason because it's complementary base pairing, and because it's complementary, the same this is exactly what will be produced. And remember that mRNA does not have thymine, is thymine, it has uracil. So let's talk about the strands, and here's the question to ask: why is the template strand, the one that is being transcribed, called a non-coding strand? So before we answer this question, we want to talk about the structure. So we have the red is the strand that is being transcribed. The transcribed strand, so either one of these strands can be transcribed, but in this example we have the red strand that is being transcribed. If the red if the red strand is being transcribed, we call the transcribed strand the template strand. We call the non-coding strand or the antisense strand. But I was asking this question. I was like, why is this called the non-coding strand if this strand is literally being translated? It doesn't really make sense, does it? But it does actually, because the opposite strand, like we just discussed a second ago, when this strand over here it will be transcribed, its code is gonna be the same as its complementary base pair, well, a complementary strand, and therefore this is called a coding strand because in actuality this strand's genetic information is actually going out into the cytoplasm for translation. So this is the coding strand, and this is called a sense strand. Yeah, that's important to recognize and understand. So now we're going to talk about the overall mechanism of transcription, how it actually happens. So actually, before we do that, let's talk about this real fast. This is and another thing to mention is upstream, and downstream. This is super important for MCAT students because we have a direct reference to promoters or termination sequences being 10, for example, 10 upstream or 10 downstream. And what really just means, if we have something upstream, it's a reference to the start point of replication. I'm sorry, of not replication. We are transcribing at the moment, but during a transcription, the start sequences. If we refer to anything above or towards the 3' end of the upstream, then it really just, or anything above or towards the other side, this side is going to be called upstream. And if we are talking about where or downstream, we're going to talk about the direction going this way, and that's going to be downstream. So upstream is symbolized by a negative sign because we're going backwards from the start point, and the downstream is recognized by a plus sign, which is going downwards from the point of origin. So with this said, we have to quickly understand why this is negative and why that's positive. Remember, upstream, you might be thinking that up is plus. That's not true. Just understand the concept understand the directionality, and you'll understand why this is plus and minus. So here is a question. Are mRNAs formed straight from transcription in all organisms? Well, we talked about this before, and we've talked about eukaryotes. And this is true for prokaryotes indeed. The transcription actually happens simultaneously, and we will talk about this later on. Don't worry about it. But transcription and translation can happen simultaneously for prokaryotes because they have no organelles, internal organelles, and therefore it's totally fine. It's acceptable. But when it comes to eukaryotes and more sophisticated cells, then they have the mRNA that's transcribed right off the bat is called immature mRNA, also called heterogeneous RNA. And we're going to talk about that later on. So the answer to this question is no. Not all organisms are right out the bat ready to use their mRNA that's transcribed. So here's the overall mechanism of transcription. So in transcription, we have 3 steps. It's a three-step process. We have initiation, elongation, and termination. And remember, we talked about the coding RNA or the mRNA having. Let's just go back to that real fast. We talked about this before. We said that mRNA has a UTR region, which is the untranslated region. We have the open reading frame over here. We have the termination code, and this really just refers that mRNA is going in the 5' to the 3' direction, and we talked about this before. And because of this, we have initiation, and remember that everything is happening in the 5' to 3' direction because, well, it's being produced in the 5' to 3' direction. So initiation is happening on the non-coding strand, and we talked about why this is the non-coding strand. So first of all, the promoter binds to the start site. So we have the promoter, which is just going to come and bind to the start site. And then we have elongation, which is when the sequence is copied off. It's just being copied off. And we can see the 5' to 3' is being constructed over here. And then we have the promoter fall off eventually when the end codons are met, and we're going to talk about that later on. And then eventually the termination is when the copying is completed and the promoter falls off, and we have the mRNA, ready to go in its next direction. Either if it's prokaryotes, it's already being transcribed or translated, and if it's a eukaryote, it has to go through more processing before it's ready to be translated.